Hello biology students, and welcome to video 5 of the organic chemistry unit. So today we're going to start our third macromolecule, and that's going to be proteins. Now proteins, it's going to take us 3 videos to get through the protein material. So if you feel like you need more information, you don't quite understand it yet, that's okay. Okay, so proteins always have 4 elements of them. They always have carbon, hydrogen, oxygen, and nitrogen. So this is different. Remember, carbohydrates and lipids just had carbon, hydrogen, and oxygen. So they always have these 4, and then sometimes they have sulfur. So that's why I've put the S in parentheses here, indicating that they always have carbon, hydrogen, oxygen, and nitrogen, and they sometimes have sulfur. Proteins have a ton of different functions, and we're not going to get to that until the third protein video, so hold off on the functions of proteins for now. Okay. So proteins, they do almost everything in your body. Proteins are like the molecule that does all the work in your body. But because they have so many different jobs, because they have so many different functions, they also have lots and lots of different structures. So this first bullet point here says that proteins are structurally and functionally diverse. One protein looks very, very different from another protein, and it would also have a different job. So this diversity comes from the fact that proteins have a lot of different monomers. Remember that monomers are the building blocks. They have a lot of different monomers that can be put together in a ton of different arrangements to build different proteins. So the monomer of a protein is called an amino acid, and that's often abbreviated AA. That's why I have AA in parentheses here. And there are 20 different types of amino acids that are naturally used to build proteins in the human body. There are more than 20 on the planet, but only 20 or so are found naturally in our proteins. And those amino acids are put together to build a polymer, and the polymer is called a polypeptide. Okay, so if each of these little circles represents one amino acid, if I had a string of amino acids, that string of amino acids would be called a polypeptide. When that polypeptide folds up, so say the string I just drew folded up into a shape, maybe something like this. Okay, once it's folded, now we call it a protein. So that difference can be confusing for some students, but just know that the polymer is called the polypeptide, and that's just the string of amino acids. And when that string of amino acids folds on itself, then we call it a protein. Okay, so let's look at what an amino acid looks like. Remember, an amino acid is a monomer of a protein. So every amino acid has a central carbon. And then branching off on one side is a carboxyl group. So we know that carboxyl group from our functional group lab last week. On the other side of the carbon, there's an amino group. And on the third side, there's a hydrogen. And then on the fourth side is what we call an R group, or a variable group. And this is what makes each of the amino acids different. Now remember I said there are 20 amino acids, so each of the 20 amino acids has a different R group. You can kind of think that the letter R is used because it stands for the rest of the molecule. There's something else there on a given amino acid. To kind of help you remember this structure, I want you to think about the functional groups that are attached to the central carbon. So an amino acid is called what it's called because the amino group here on the left gives it the amino part of its name. And then it turns out if you put a carboxyl group on something, it makes it more acidic. So that's where the acid comes from. So you have an amino group and a carboxylic acid group, which is why it's called an amino acid. Okay, so let's look at a couple of these R groups, so just so you can get an idea what they look like. Do not memorize the different R groups for different amino acids. I just wanted to show you how the R groups differ. Okay, so for example, for this amino acid, this monomer is called leucine, and you can see it has this R group, a branched carbon R group. And it turns out that this group is hydrophobic. So if a protein has a lot of leucine monomers, that protein is more likely to be hydrophobic. Similarly, serine has this R group, aspartic acid, a different amino acid, has this R group, and these R groups are hydrophilic. So if a protein has a lot of these R groups, the protein is more likely to be water-loving or hydrophilic. So basically, these R groups kind of help to dictate the properties of the protein that they make up. Okay, So like we already know from carbohydrates and lipids, we're going to join amino acids, the monomer, together using dehydration synthesis, okay? So for example, I have 2 amino acids right here. If I take away this water, I'll form a bond between this carbon right here and this nitrogen right here, okay, to form what we call a dipeptide. So a dipeptide is kind of like a disaccharide. It's two amino acids together, and I've also removed and formed a water. Now it turns out the bonds that link amino acids have a special name called peptide bonds, okay? So the bonds formed by dehydration synthesis of 2 amino acids, that bond is called a peptide bond. And that bond is always between a carbon and a nitrogen from the carboxyl group and the amino group on the 2 amino acids. Okay. So we'll talk about a couple more examples of this process. So here I have 2 amino acids. You can see they have different R groups. They're different amino acids. I take away a water, okay? There's actually supposed to be a hydrogen here. That's confusing. So I take away this OH and that hydrogen, okay, and end up forming a bond between this nitrogen and this carbon to form this peptide bond, and now I have a dipeptide. So here's a more complicated example. Here I'm linking 1, 2, 3, 4 amino acids together. I wish there were hydrogens drawn on these. That's confusing. Okay, so I take away this water, I take away that water, and I take away this water right here. Okay, so I take away 3 water molecules to form 3 peptide bonds. So as you can see, what I formed here is a polypeptide, 3 or more amino acids linked together. So that's it for video 5, and I'll see you in video 6.